All right, so now that you know the vocabulary, if you don't, watch that video, or if you already know it, you don't need to, but then we're going to talk about what you can do with that. All right, so one of the things that comes up a lot is called monohybrid inheritance or Punnett squares. So this is the idea that, okay, so let's say you have, again, a blue-eyed and a brown-eyed person, all right, and you want to know what is the chance of them having a blue-eyed kid. All right. Now that depends on their genotypes. So now we know that brown eyes, excuse me, is dominant. So if you have a brown-eyed person, they could have two possible genotypes, right? They could have this, or they could be heterozygous. So they could be homozygous dominant, or they could be heterozygous. You don't know. Okay. And then you have, but if you have a blue-eyed person in our perfect world, right? Remember, there's only this monohybrid, then they have to have this genotype. They have to have two small b's. All right. So let's say, for example, let's move away from humans. All right. And let's say these are mice. Just pretend with me here. All right. So we have a brown-eyed mice, and we don't know what its genotype is. We just know it has brown eyes. But we have a blue-eyed mouse that we can cross it with. Okay. This is called a test cross. We're literally going to breed these mice and see. Now, what you would do is say, okay, I'm going to take this brown-eyed mouse, and I don't know what its genotype is, but I want to. I don't know why, but you do. And I'm going to cross it with this blue-eyed mouse. All right. So how do you do that? You draw a Punnett square. So the idea is that the blue-eyed mouse, okay, is always going to all of her gametes are going to have two small b's. That's it. There's no other option. All right. And you're going to need to do this twice. So the idea is that the first Punnett square, let's assume that this is the genotype. All right. So I'm going to do one big B. So each parent can only give, this one can only give a large B, and this one can only give a small b. So the idea is that, so these represent the gametes, because remember that gametes only have 23 chromosomes, so they have either this or this. They don't have both. And the same thing here. These are the gametes as well from the boy. I don't think I ever said which one was a boy and which one was a girl, right? And so then the idea is, okay, well, what are then, you fill in the squares. It's like, I don't know, multiplication, but not. Okay, so it's BB, right? BB, BB. Now I'm just saying BB too many times. All right, and so the idea is that if this is the genotype of the brown-eyed parent, all of the offspring you can see are going to be heterozygous, and they will all have brown eyes. 100% of these offspring will have brown eyes. All right. Now, but what if that's not? What if this is the genotype? Okay, well, let's see what that would look like. So the recessive, the one with blue eyes, doesn't change. That still offers too small. Okay, but now the idea is that the brown-eyed parent, could give either a big B or a little b. And then you fill it in. Aha. See, now what you can see is that there's four possibilities, right? There's always four possibilities in a monohybrid inheritance. There's four possibilities, but two of those four would give you brown eyes, and two of those four would give you blue. So that means that instead of all having brown eyes, you'd have 50% have blue eyes, and 50% have brown eyes. Now, this is a possibility. So you might not end up, if the mice have 20 kids, you might not have 10 with blue eyes and 10 with brown eyes. You might have 9 with blue eyes and 11 with brown, because this is a possibility. It doesn't mean that it's going to be evenly split, but you should still be able to know. The idea is that it's called a test cross, because you cross the unknown genotype with a recessive genotype, and then based on the offspring, you can determine what is the genotype of the parent, all right, of this original parent. All right, so this is called a Punnett square, this idea, and you can do it for anything. And so if they give you, heterozygous or homozygous, and they want to know the offspring, the idea is that you talk about the chance. Now, in order to get full points on this, all right, then you need to do this. You need to spell it out. You can't just do the Punnett square and leave it and say, oh yeah, it's obvious. All right. You have to spell it out. You have to associate which of these phenotypes goes with these, which of these genotypes go with the phenotype. So for example, here I would say, oops, I wouldn't say that. This is BB, and I would say this is the big and little b, so that that you're showing the examiners, you understand that this is the one that gives you brown eyes, and this is the one that gives you blue eyes. All right, this one you would say They're all going to have brown eyes based on that. So that's a test cross using a Punnett square. All right. And the Punnett squares come up a lot anytime you're talking about monohybrid inheritance. This sounds fancy. It's not. Monohybrid inheritance literally means that only a single gene is controlling a phenotype. All right, which is not true for many different things. In plants, you'll often get it as well, right? Different colored plants or things like that. So that's a Punnett square with monohybrid inheritance. I'm going to talk about codominance in a another video. So this is all about recessive alleles. Yes, okay, recessive and dominant alleles. Then you talk about a test cross or monohybrid inheritance. When you have codominant, it's another ball game, and I'll do a video on that.